I think I will get started. Today, well, first of all, thank you everybody for joining. Today I'm pleased to introduce Matt Fitzpatrick for a seminar related to his promotion process. I'll just briefly give a little background about Matt. He got a bachelor's degree in mechanical engineering at Penn State, and that was a bachelor's degree, a master's degree in environmental science from the University of Montana, a PhD in ecology and evolutionary biology from the University of Tennessee. After that, he went on to a postdoc position at Harvard Forest and the University of Rhode Island. And then he came to the Appalachian Laboratory in 2009 as a tenure track assistant professor. He was promoted to associate professor in 2015 and has been in that position since. He did a sabbatical as a distinguished visiting researcher at the CSIRO Land and Water Institution in Canberra, Australia. He's also received a number of awards while he's been at UMCES, including the UMCES Professor of the Year in 2013 and the UMCES President's Award for Science Application in 2019 and the UMCES President's Award for Outstanding Faculty Mentorship in 2020. So I'm going to keep it very brief to the point there and hand it over to Matt. I will remind you, however, that this is being recorded so that others who are unable to attend today will have an opportunity to view the presentation. And also that there are still a few sign-up slots, and we could always make more sign-up slots if needed for those of you who would like to meet with Matt. And if you don't have the link or know how to do that, you can get in touch with Rhonda Schwinebart or myself. So with that, I'd like to hand it over to Matt.
Okay, thanks, Eric. Let's try the screen sharing here, and everybody sees my slides, right? Good. Okay. So thanks everybody for tuning in. It's great to have the opportunity to present to colleagues. We don't get to do that a lot, so it's definitely a special opportunity when it arises. And so I thought I would start with a quick trip down memory lane to get things started here. So what I'm showing you is the title slide of the first talk I ever gave at UMCES at the Appalachian Lab about 12 years ago in November of 2008. So this is the my title slide of my job talk. And you can see from my title, I was thinking then about the things that I do now, which is modeling the responses of species to climate change. And I list adapt, migrate, or go extinct as the header here on my slide. And that's alluding to the idea that when organisms are faced with climate change or environmental change, they sort of three outcomes or choices, if you will, in response to environmental change. They can stay where they are and attempt to tolerate those conditions, or perhaps over longer timescales, adapt to the new conditions. They can migrate elsewhere to another place where suitable conditions are present and establish in those places. And if neither of those are an option, they could possibly face either global extinction or local extirpation. And had someone asked me on my interview, and I don't think anybody did, like how would I actually combine these in a modeling framework, it would have been a tough question to answer because at the time, the tools didn't really exist. And the data that we need to answer these kinds of questions, especially from the adaptation angle, were not necessarily in their infancy, but certainly not widespread. But I feel like we've made a lot of progress in my lab on this topic. And so that's going to be the bulk of what I talk about today. And so the first part of my talk is is going to focus on this idea of doing kind of comprehensive climate vulnerability assessments in forest trees, incorporating genomic information into these models to get a sense of the interplay between adaptation, migration, and extirpation of populations. Second major thrust of my research is model evaluation. I mean, the nice thing about making forecasts to 2080 is that I'll be dead before anybody can prove me wrong. But I do think it's really important that we test these methods that we're using to make predictions, right, or forecasts. Anybody can make a forecast, but the important point being that these models and methods that I use a lot are being used to inform decisions and policy. And so we need to know how well they're going to work. And if you're thinking about models at the species level, it's really hard to do experiments with species ranges. And so often I've looked to the fossil record as a means to try to do some evaluations of these methods. But when you work at the level of populations, things get a lot easier because you can actually move populations around and do experiments on them. And so that's what I'll talk about in the second part of my talk. And then I'm gonna, for the last part of my talk, move away from natural systems and talk about how we can use some of these spatial modeling methods to communicate climate change impacts in cities. Okay, so part one, building on this adaptation, migration, or extirpation idea. So for this, I'm going to use red spruce as a motivating example for a number of reasons. This is a red spruce forest in West Virginia. It's at the top of aptly named Red Spruce Knob. And I think these forests are some of the most incredible and interesting forests in Eastern North America. In the central and southern Appalachians, they only occur at the highest elevations that stay cold enough and stay damp enough for red spruce to thrive. And so within the Appalachians, this is an imperiled ecosystem. It's been threatened for some time. Historically, these forests were heavily logged. They were also impacted by mining, and they were also impacted by acid rain. And so there's a lot of interest in restoring these forests. Efforts like the Central Appalachian Spruce Restoration Initiative, or CASRI, has been planting hundreds or thousands of red spruce seedlings across West Virginia to try to restore these forests that provide important ecosystem services, including habitat for wildlife. But red spruce is in trouble. This is some projections from work that postdoc in my lab, Susanna Lockmuth, has been working on. And you can see this model is predicting a substantial shift in the suitability of climate for red spruce, so that the current projection is here on the upper left. We see that by 2090, that has shifted to the north, which means the climate suitability throughout much of the range in the US, and certainly the Southern Appalachians, is going to decrease to the point that red spruce probably will not be able to exist in many places it does at present. And so if you're a CASRI and you're interested in restoration, you might be asking yourself, are we wasting our time? Or perhaps how might we try to confer resilience so that the individuals we're planting have the best chance of surviving, at least for the near term? And that sort of question isn't one that these kinds of methods can answer. We can make these broad scale predictions of range shifts, but migration and the movement of the species range is not what's going to be the major factor over the next several decades. It's going to be the ability of populations to tolerate these changes in place, and potentially maybe some hope of adaptation over longer terms. And so as part of a project, collaborators and at AL, Dave Nelson and I and folks at the University of Vermont and the US Forest Service have collected red spruce seeds across the range of red spruce. And we've grown up those seeds in a greenhouse, and there's Dave and I leaving the University of Vermont with a trailer full of about 2,000 red spruce seedlings, which we planted in a common garden outside the Appalachian Lab. And you can see in this photo that the seedlings were roughly all the same size when we put them in the ground. To this point, they had only lived in a greenhouse. This experiment was harvested just a few weeks ago. A big shout out to Dave and Susanna and Natalie and Erica, who did this work while I was quarantining from COVID, so I didn't have to do any of the back-breaking work. But when we harvested these individuals and measured their final heights and things like that, you can see that there's a lot of variation, right? There were trees that grew to over 1 meter in height. Natalie's holding one of the larger specimens here. You can see the long root in her hand. And some remained, a few centimeters tall throughout the duration of this experiment. So this is individual variation, and that individual variation driving how these populations and individuals are responding to the climate in the common garden outside the Appalachian Lab. And so one of the questions I've been really interested in is, how do we get this individual variation into models in a meaningful way? And so to answer that question, I'm going to pick up basically where I left off in my tenure presentation. I guess that was about 6 years ago now. At that time, working with Steve Keller, I came up with this idea that some of the models that I had been using to understand and predict change at the level of assemblages, so communities of species, might be an ideal way to model some of the data that Steve was working with, which is genomic information within species. And so we proposed this idea in a paper to Ecology Letters. And for that work and the work going forward, we worked with a species of tree called balsam poplar. And so I often get asked the question, well, why balsam poplar? Well, and the answer is mainly because it's closely related to black cottonwood, Populus trichocarpa, which was the first tree to have its genome sequenced. And so balsam poplar is not that far from a model system. And so it was kind of ideal for developing some of these methods, but using natural systems. So this paper has generated a lot of interest, and so we've been excited by that. But here's the basic idea of how this modeling goes. So this is the range of balsam poplar. It spans these really large gradients from northern Canada all the way to the Appalachian Mountains in Pennsylvania, and also some pockets in the Rocky Mountains. So we have these populations distributed across the range of balsam poplar. So the basic unit that we're interested in are alleles, or single nucleotide polymorphisms, or SNPs. And so, these, if we went out and sequenced these individuals, we would find that their underlying genetic sequence is going to vary across the range of the species. And so the northern population might have an allele, a frequency of alleles such that allele 1 occurs in about 10% of individuals, and allele 2 occurs in about 90% of individuals. And we would look across all of the individuals we collected, and we might find that the southern population has sort of the reverse. 90% of the individuals have allele 1 and 10% have allele 2. And so it's this spatial variation in the genome that underlies local adaptation. So one of the big challenges here is that there's variation in the genome that doesn't underlie local adaptation. It's just neutral. And so parsing those two things is a lot of the work that goes into this, but I'm not going to say too much about that. And so one of the methods I had been working with is known as gradient forest, kind of an unusual name, but it's in essence random forest. It's based on the random forest algorithm, which is used in all sorts of different contexts. And gradient forest is in essence just extracting some of the additional information from random forests that normally is not used directly. And so using this method called gradient forest, we can model this genomic data where we have alleles as columns and we have populations as rows. And we might be dealing with a few dozen alleles, or we might be dealing with 100,000, depending, or SNPs, I should say. And so one of the main outputs from this method are these turnover functions that I'm showing you here. And it's kind of important that we understand these, because a lot of what I'm about to show you is going to be based on these. And so this is a turnover function for a set of adaptive SNPs within balsam poplar. And we're looking at how that signal varies as a function of mean summer temperature. And so there's two kinds of information that we can get from this function that are relevant here. One is the overall shape. It tells us something about the rate of change in allele frequencies along each of the gradients that we fit in this model. So in this example, there's not much change in allele frequencies along the coldest and warmest end of this gradient, but there's a very rapid change in allele frequency in the middle of this gradient. And so that is suggesting that adaptation to climate is driving variation within the individuals, mainly in the middle portion of this gradient. The overall height tells us something about the relative importance of that variable and explaining the variation in the genomic data, with greater height meaning greater importance. And so overall, we can look at these functions and get a really good sense of not only how the genome is varying, but what's driving that variation in terms of environmental predictors. So the other thing that Steve Keller and I proposed was this idea that we could use these functions that are fitted in space to make predictions in time. So this is the classic space for time substitution that's often used for understanding how systems might respond to change. And so to understand that, we can imagine having a population A that falls at some point along this gradient and a population B that falls somewhere else on that gradient. And so if the climate were to warm, for example, population A would warm up so that it's now warmer in that location. And the genetic offset is then this distance between where population A falls here and there. So this is like the amount of genetic change that would be required for A to have the same relationship with climate as it does at present. And so we get a big genetic offset for A. Since B falls along the flat portion of this curve, the same amount of climate change is going to produce a smaller offset, because individuals along this part of the gradient don't show much variation in this particular segment that we're modeling. And so, we can use this approach, this space for time substitution approach, to make predictions of basically maladaptation. So if a population is exposed to climate change in place, how maladapted is it going to be in response to that? But if we want to get at these other components, we want to think more broadly about these other possibilities, we need to go beyond this sort of static in situ thinking. And so working, thinking more about this after this paper was published, and working with my grad student, Andy Gowerty, this is what we came up with. So imagine you're hiking in the woods and you enter a stand of red spruce or aspen or whatever your favorite tree species is, and you stop there for a moment and you think about, what would happen if climate changed, right? And so we're staying in the same place and we're imagining climate changing. And so we could think about how much stress would that population experience given the change in climate. That's what I just presented to you. That's what we called genetic offset originally, and that Andy and I rebranded as local offset because it's assuming the population stays in place. And so that answers the question, how maladapted would a population be given its exposure to climate change in its current location? We can also define other possibilities. We can ask, well, what if we could move this population anywhere and how maladapted would it be to those conditions, right? So we have this population at time 1, at place 1, and we say we're going to move it anywhere at time 2 and calculate what the maladaptation would be there, or the offset would be there. And so we could move this population anywhere on the landscape, and we could find the place that minimizes maladaptation. So like, where's the place we'd want to move this to ensure this population exists in the future? And we called that forward offset. And that answers the question, how maladapted would this population be if it moved anywhere else, or we moved it or its genes anywhere else? The other possibility is a little bit harder to get your head around. But imagine we were in the same place, but in the future climate. We could ask, are there any current populations on the landscape that could move to this location and exist? Or if they did move to this location, how maladapted would they be? So for that, we look only within the current range of the species, or only in places where we know populations reside. And we calculate what we call the reverse offset and find the minimum value. And that answers the question, are there pre-adapted populations out there, and how pre-adapted are they? Okay, so thinking back to my job talk 12 years ago, we get closer to this idea of integrating all of these different possibilities by considering these various kinds of offset: local, forward, and reverse. Local being in situ vulnerability, assuming no movement or migration of individuals. Forward offset being the possibility of extirpation, assuming migration, which you could limit to some distance if you wanted, if you knew something about dispersal. And then reverse offset is this pre-adaptation to future climate. And so we applied this idea again to the balsam poplar system. And we wanted to know basically, where's the greatest vulnerability? And that's what these questions are getting at. So where will climate change cause the largest mismatch between climately adapted populations and climate? Which populations may be least or most adapted to future climates? And where will populations have to migrate if they want to minimize migration? Or where should we move them to minimize migration? Or maladaptation, sorry. So for this study, just quickly, we were working with 81 populations. So we had a comprehensive sample across the entirety of the range of balsam poplar. And we focused on 75 SNPs within the flowering time gene network. Those control phenology in balsam poplar. So when do leaves come out, when do leaves fall off, when does the tree stop growing? And so these are important for determining how much biomass the individual puts on throughout the growing season, but far from comprehensive. So luckily this method produces really intuitive graphics that require no explanation whatsoever. And so I'm just gonna move on. So it's really hard to tell jokes on Zoom because you can't hear anybody laughing, but maybe no one ever laughs at my jokes. That's also possible. Anyway, so this is the kind of output that comes out. And what, it's hard to show something as multidimensional as all of these different factors and their relative importance. But the idea that Andy and I came up with is we can assign each of these kinds of offset to either the red, green, or blue channel. And so we can produce an RGB image that shows the combinations and the relative importances of these different kinds of processes. And so in this figure, the dark shaded regions is where are where all of the offsets are low. So this is where there's low local exposure to climate change, potentially because climate's not changing that much in that location or other reasons. Individuals or populations are pre-adapted to future climate elsewhere in the range. So that's low forward offset. And there's potentially existing populations that are pre-adapted to these conditions, either locally or elsewhere in the range that could move to that location. And so we get low vulnerability relative to everywhere else. The light shaded regions are just the opposite. It's the maximum combination of all these possibilities. So it's where we see high local exposure, so that's high local offset, there's no place that you can move those individuals to avoid extirpation. So that's high forward offset. And there's no existing populations that are pre-adapted to these conditions, possibly because these climates are beyond what the species occurs in its current range. So the other really, I think, informative thing that we can do is we can ask, well, how far do we have to move these populations, or how far do these populations have to migrate to minimize their vulnerability? And we see that that varies a lot across the range of the species. In particular, in the northeastern portion of the range, these individuals would need to move on the order of 4,000 or 5,000 kilometers. And actually the locations you'd need to move these to minimize maladaptation is on the westernmost portion of balsam poplar's range. We can also ask, well, what direction do these need to move, right? When you think about, at least in terrestrial systems, and this is probably true in marine systems as well, that when temperature goes up, we sort of would assume this poleward march of species ranges, right? But the actual data suggests it's not that simple, at least in the near term, that populations are often doing things slightly different than that. And this sort of bears this out when we take this population level look at things. We see, of course, a lot of green here, which is vectors moving to the north and the northwest. But there are places where populations moving to the south would minimize adaptation, and east and west as well. So we see a much deeper assessment of how populations might respond to these changes. And so this question about adaptation, migration, or extinction is actually one of adaptation, migration, and extinction, right? It's all of these occurring to various degrees in different places, depending on the circumstances. We can identify where within the range processes like migration and adaptation may be most effective at reducing the maladaptation under future climates, which is exactly the kind of information I think that if you're trying to do restoration or conservation in an age of rapid environmental change that you want. And it also could inform assisted migration. So which populations should we move, or which species should we focus on, as well as assisted gene flow, if we want to think about trees and moving genes around or not. Okay, so just this morning, the proofs for Andy's paper arrived in our email. And so if you want to read more about this, this should be out soon in Nature Climate Change. And we're really excited in general, but I'm really excited for Andy to have landed a paper in a journal of this magnitude in his dissertation. And hopefully he continues to do really great stuff. Okay, so that was part one. So part two is this question about, okay, Matt, you can make these pretty eye candy maps. Do they really mean anything? Are these methods going to work or not? And so we can nicely test this space for time substitution by performing an actual space for time substitution with populations. So in this case, we're taking balsam poplar populations from across the range and transplanting them into common gardens in Burlington, Vermont, and Indian Head, Saskatchewan, and measuring how well they grow, measuring their phenology and the timing of leaf out and that sort of thing. And so we have a couple of questions here that we're interested in. The first is, well, do genetic offsets predict performance of populations in the common gardens or not, as we hope they do? And the second question is, do they do better than just using naive climate distances? And by naive, I mean, we can just measure the difference between the climate where a population grows and the climate at the garden, and use that to predict how well individuals do. And if these genetic offsets are valuable, we would hope that they should do substantially better. And then the third question is this going back to this idea of actually isolating the portion of the genome that's underlying local adaptation from just the background signal. And so the prediction here is that genetic offsets based on what we would call candidate SNPs, those actually associated with local adaptation, or at least that we hope are, should predict performance in the garden better than just SNPs selected at random from the genome. And so for this portion of my talk, we're focused on 42 individuals from just the center of the range. We're trying to limit the influence of population structure and focus on these populations from the core of the range. But we had much more comprehensive sequence data here. So we're working with over 100,000 SNPs that are going into these models. And so here are the turnover functions, and there's just a few things that I want to point out here. So, you know, I mentioned this idea of candidate SNPs, the SNPs that are associated with local adaptation. There's a lot of, this is a very active area of research, and there's several different methods available for trying to determine which SNPs are locally adapted or not. And we used a number of different methods for this study, and I'm not going to go into those details here, but I just want to point out that the curves that don't have kind of the black lines around them, those are different sets of candidate SNPs identified by different methods that we're fitting gradient forest to. This cyan line here with the black lines around it, this is the cyan line is the mean of 999 models, or I'm sorry, it's models fit to 999 random draws of 500 SNPs. So this is the background signal, and each of these black lines is one of those models, and this is the mean. Hopefully that's clear. And then these are the curves for the candidate SNPs. So latitude came out as the most important variable overall, driving variation in genetic patterns for these 42 populations. The second most important variable was winter temperature, and you can see the rest of the variables here. There's a couple of things I want to point out. So this population here, which I haven't told you yet, these three-letter codes are the populations and where they fall, in this case along latitude. CLK is the most northern population, highest latitude. And so the offset of planting CLK in either the garden in Saskatchewan, the location of which is shown as this dotted line, or the Vermont garden would be the change in height from where CLK falls along each of these curves and intersects this line. And so, you know, moving CLK to the Vermont garden would have the largest offset of any population that we tested. Okay, so I want to show you these results first in geographic space. So what I'm showing you is a map of the 42 populations, and the size of the dot is scaled to the predicted genetic offset of transplanting this population in the common garden. So the larger the circle, the worse we expect that population to perform in the experiment. So we see this pretty clear climb that goes from the northwest corner towards the garden. And I just want to point out this one population here. This is Shoshone River outside of Yellowstone. Has a sort of a middle of the road offset based on the size of this circle. And here's Stephen Gwynn and I collecting balsam poplar along the Shoshone River several years ago now. So that's the genetic offset pattern. The climate distance pattern is much different. We still see, I should say too, this is that population CLK, it's the most northern one, has a high climate distance, but you can see this doesn't really follow the pattern predicted by genetic offsets. So climate distance and genetic offsets are telling us two different things. Why is that? Well, we can move from geographic space and look at where these populations fall. in climate space just by doing a principal component analysis on these different climate predictors. So the shading here are different grid cells within balsam poplar's range, and the labels are where the populations fall in this multi-dimensional climate space. And the triangle is the location of the Indian Head Garden in this space, and the circle is the location of the Vermont Garden. So the naive climate distance is just the Euclidean distance between each population and the common garden. And so you can see Shoshone River is way down here because it's a very high elevation population. It's unusual in that way. And so this has a really large climate distance because it's falling really far from the Vermont Garden in this space. Here is that same space, but this is the output from the gradient forest model. And so now we have properly weighted these different predictors to how they are driving the underlying genetic pattern. And so I said latitude was the most important predictor. You can see this is heavily loading along this axis. And then winter temperature was the second most important variable, and it's loading most heavily along this axis here. And so this space is now scaled to the genetic patterns. And so genetic offset is just the Euclidean distance in this space between each population and the common garden. And we see Shoshone River falling a lot closer, right? Because we've now corrected for the genetic pattern, and we see that it has a much lower offset than it does climate distance. Okay, so let's get to the important part, which is, does this work? So naive climate distance here explains about 34% of height growth in the common garden. The model R squared was about 0.34. Genetic offset, in contrast, based on candidate SNPs, right? So these are the SNPs underlying local adaptation, explained about 63% of height growth in the common garden. So they're doing, the genetic offset is doing much better than this raw climate distance. So there's useful information that we're extracting from that. But here's the surprise. The genetic offsets based on random SNPs did just as well or even slightly better in explaining height growth in the common gardens than did the candidate SNPs. So we do all this work to try to figure out the SNPs that are underlying local adaptation. And in this experiment, we could have done just as well, if not better, by just using the random background signal. And so, why is that? We have a couple of reasons. The most obvious being is that the background pattern of genetic variation is nicely aligned with the adaptive signal, which makes it a challenge to tell the difference between these two. But it means that the background pattern contains useful information for predicting performance in a new environment. But why the candidate SNPs don't do better is a little bit of a mystery still. I will say though, I have another collaboration going with Katie Lauderhouse at Northeastern University, and Katie is a real whiz at developing evolutionary simulations. And so we've tested these methods using simulated data, and in that case, we actually know the loci that are driving fitness, that are underlying local adaptation, and we see the exact same pattern. We see the neutral signal doing just as well, if not better, than the adaptive signal. So some head scratching there yet to be done. Okay, so answers to the questions. Yes, genetic offsets can predict performance, and they can do so better than naive climate distances. But at least in the work that we've done so far, candidate SNPs don't do any better than randomly selected background. Okay, so I think I left that slide in there by accident, so I'm gonna skip that. Okay, so I'm gonna move away from balsam poplar and forests and talk quickly with the time I have left about using spatial modeling methods to communicate climate change impacts in cities. And you might be asking yourself, why does a guy that thinks about terrestrial systems and forests care about cities? Well, there's a couple of reasons. The first is that over half of the world's population lives in urban environments, and urban environments are highly sensitive to climate change. Of course, these places contain a lot of infrastructure to keep people happy and healthy, and that infrastructure is sensitive to extreme events and other aspects of climate change. And so it means, a lot of people are potentially threatened by the changes we anticipate. The other reason is it's really because there's so many people in urban areas, it's important to communicate to them about the risks of climate change. And as you probably know, probably half or a little less of Americans are not as concerned about climate change as we think they should be, or they're not concerned at all, especially compared to scientists like us. And so there's lots and lots of reasons why that's the case. But I think a contributing factor is how we communicate climate change to the public. And so what I'm showing you here, which is this example I like to use, is on the right is a screenshot from my phone showing the weather forecast for Frostburg. And you can see that the daily highs and low vary by several degrees Fahrenheit. And that daily variation exceeds the changes in mean global temperature shown in these coarse maps, and also the 1.5 degrees C limit that we are concerned about exceeding and going into a state of potentially irrevocable damage. But if you're a member of the public, these numbers, 1.5 degrees C, or your hometown is going to warm by 5 degrees Fahrenheit, doesn't really mean much. And it's hard to sort of articulate what those changes mean. So that was one of my major motivations for performing this work. And so to get around this challenge and to kind of bring this information to people in a way that's more personal and more local, we can use a technique called forecasting by analogy. So instead of just saying, well, what will Washington, D.C.'s climate feel like in 2080? Summers are going to be 8 degrees Fahrenheit warmer and 30% wetter. We can say, or ask the question, is there a location in North America that has a climate today that is what we expect Washington, D.C.'s to be like in 2080? Where is that place? And maybe people have some relationship or at least some broad knowledge of climate in that place to better understand the magnitude and rate of change we're expecting. And so here's a map showing the climate similarity for Washington, D.C.'s climate in 2080. There's a couple of things I want to point out. The yellow means that's a good match. That's a good current match to Washington, D.C.'s climate in 2080. So if you want to know what Washington, D.C. is going to feel like in 2080, go to one of these places shaded yellow that I've outlined in a bold line. Once you get beyond, I forgot to say that the values I'm matching here or mapping here are called sigma dissimilarity. And so it's a metric of how similar Washington, D.C.'s climate in 2080 is to all of these locations. Once you get a sigma dissimilarity greater than 4, you're really talking about a poor analog and what we would call extreme climate novelty. So what we're saying is there's no place, if you have a sigma dissimilarity greater than 4, there's not really a place that's a good match at all. So for this example, this is under reduced emissions, what we would call RCP 4.5. We see that the best match in this yellow area here falls near Paragould, Arkansas, and it's a sigma of about 0.57. So that's a pretty good match. It's less than 2, which we consider to be the limit for a good analog, and certainly less than 4, which we consider poor analog or extreme dissimilarity. Under high emissions, we see this map kind of shifting to the south and also getting darker, because as emissions go up, climate in Washington, D.C. changes to the extent that there's not really a good analog anywhere in North America anymore. We're still under 4, but we're above this value of 2. And so the best location for Washington, D.C.'s climate in 2080 under high emissions is Greenwood, Mississippi, and we get a sigma of just under 3. So we performed this analysis for about 540 cities in North America, and these are the vectors showing where all of the cities' analogs are. This is under high emissions. And the take-home point here is that if you're an average dweller, urban dweller, and you want to know what your city is going to feel like in 2080, you should go about 800 kilometers into the south to find that place. In terms of matches for like how good are these analogs, for low emissions, we find about 70% of cities have a good match within North America. So we can find a place that has a pretty close match to that place's climate in 2080. However, under high emissions, we find increasingly poor matches, and there's very few, I think it's under 20% of cities that we can find a meaningful match for in North America. And so what this means is that under high emissions, most cities that we analyze are going to have a climate that is not like anything currently present in North America, which isn't good. So we, of course, built a web application that you probably heard about. I'm not going to demonstrate this today, but if you haven't looked at it yet, you can go to this link, or you can go to the UMCES website and find it from there. But preparing for this talk was a good motivation for me to finally go and look at the hits that the application received early in its release. And that's what I'm showing you here. And so the embargo was lifted and the paper came out at about 4 o'clock on Tuesday. I think it was February 12th, I think the date was. And you can see that the visitors rapidly ramped up, exceeded about nearly 300,000 within the first 2 hours, which promptly killed the AL server, and the app went offline. It was off for a while until we were able to find someone else that could host it. So within those 2 hours, we had hits from all over the globe. Most of them were within the US, but in all sorts of far-flung places, people were checking this out. I received an email from National Geographic, and they wrote me and said, you know, what are your future plans for these analyses? And luckily, I was smart enough, not always the case for me, to realize this was an opportunity. And I replied and said, I have all these amazing ideas, I just don't have any money to do them. And so they said, well, we'd like to fund you to do a global analysis for our Earth Day 50th anniversary special issue, which came out right about the time that the pandemic hit. And so I think in large part, this issue was actually a flop because no one was paying attention. Which is too bad, but it was still fun to have the opportunity to do that. So for that analysis, we looked at about 2,000 cities globally, and I don't have the time to show you the results. And honestly, it's kind of more of the same idea. But there's two things that I want to show you quickly. The first is I was curious about, would we find a better match for places like Washington, D.C. if we looked globally? So for the first analysis, we only looked within North America. But if you look at more places, chances are you're going to find a better match. And so I was curious, to what extent was that true? So it turns out that if you look globally for Washington, D.C., and this is actually true of a fair number of cities in the eastern US, the best match is in the foothills of in Pakistan. And we actually find a really good match. So this is year 2070, slightly different than the 2080 stuff I presented earlier, but that was the date that National Geographic wanted. But we find a really good match, the sigma of 0.12. And so this is a really different place climatically and in terms of the vegetation and everything else in that area. So it's pretty substantial change that these models are predicting. But it turns out that if we look only within continents or we look globally, there's not large differences overall. Most of the differences are small, and so I don't feel like I overestimated the degree of mismatch in the original study for not searching globally. And so lastly, since we're looking globally, we can ask this question of what is the exposure of cities to climate novelty? And so what I'm mapping here is out of the 2,000 cities, those that show extreme novelty. These are the cities that don't have any modern match to their future climate. Not surprisingly, these are clustered around the equator, right? Because a lot of these places are becoming hotter than any place at present. And what's frightening is just within these dots, there's about 400 million people that are going to experience extreme climate novelty by 2070. And many of these cities are in places that don't have infrastructure to handle the burden that this is going to place on these people. And so if you think the pandemic has caused a large disruption, I think it's going to pale in comparison to what we might see as climate change ramps up. And so with that cheerful note, I will stop there and thank everybody once again and be happy to take any questions.
Sorry, had a hard time unmuting. Thank you, Matt. If you could stop sharing, well, maybe you should continue to share your screen, I suppose. It's probably a worthwhile thing. So I'll do my best to try to look for people who have indicated that they have a question, but it is kind of hard to do with with Matt's screen still showing. So if
I'll stop sharing, Eric. I can always switch to a slide if I need to.
Okay. Let's see. Andrew, why don't you go ahead? I would suggest you do as Andrew has done and put your name in the chat box, and I'll call on you.
Hi, Matt. Sorry for the naive question, but how confident are we that genetic distance is correlated with the probability of it happening? So my understanding is that that requires either a long distance dispersal or evolution to meet that genetic distance to bridge that gap. And I don't know how correlated it is with the probability that that is actually something that could happen for that population.
Yeah, I mean, I think the flip side of that is it's not likely to happen. And so that's why we can interpret this as a vulnerability. And so it's kind of, you could think of it as an adaptive stress or an adaptive deficit that these populations are going to experience. And as in terms of how rapidly might that happen, I mean, that's a very widely from organism to organism, depending on generation times and what have you. But over the next several decades, especially for long-lived organisms like trees, it's not going to happen at all. And so we're going to have to decide whether we want to move things around or not. And I think that's where these methods can at least help inform that somewhat.
Okay, Tom, and Matt, I'm not sure that you actually stopped sharing your screen. I think you went off of presentation mode, but
well,
anyway.
Is it okay if I keep this up, and if somebody
sure,
needs a
that's
slide,
fine.
I can get to it?
Sure. Tom, please.
Thanks, Eric. And Matt, thanks for a very clear presentation. So I work in an environment where if the organism doesn't like the climate, it moves away, right? You work in an environment where the trees don't have that choice. You also work in an environment where the life history of trees are arguably probably longer generation time than many of the fishes, at least many of the exploited fishes that are left have. So I wonder whether you would talk a little bit about the interaction between generation time, the rate of climate change, and our ability to see responses.
Yeah, I mean, the way this was originally envisioned was in trees. And when we defined a genetic offset in that paper, we said this is most relevant to long-lived organisms that can't move, because ones that can rapidly disperse are going to probably have that as their primary response. But even if you move, even if organisms can move to a new place and potentially find suitable environments, but if I'm thinking like in the context of restoration, right? Like we may want to keep some of those organisms in the same place as long as we can, and so in that case, I think there would be useful to identify adaptation. And then there's also the problem of genes moving around that. Especially in trees that exchange genes a lot, that they can be limited in their ability to adapt just because the genes are moving around so much. There was a paper that came out in Science, I guess about maybe 2 years ago now or so, that was using this for birds, to look at climate change impacts on birds. And we felt like it was inappropriate because birds can disperse really well, and probably it's not so applicable in that context. So I think it does depend a lot. You asked the question about generation time too, that trees can live centuries, and so a lot of these effects are going to take centuries or decades or centuries to play out. So, sorry, did I answer your question, Tom?
Yeah, you certainly got at it, and we'll pick it up next week, I think, as well. I'll look forward to the discussion.
Okay, I'm going to call on Claire next.
Okay. Yeah, thanks for the great talk, Matt. It was like really super fascinating research. I was wondering about kind of similar to what Tom was asking about generation time, and when you're collecting seedlings to grow or seeds to grow in the common garden experiment, are they from populations that are like stable and viable in terms of like, I could imagine situations where you have a mature forest that aren't necessarily, there's not a lot of active recruitment. And so you're measuring conditions, climate conditions, and saying this is a viable population, but maybe the climate conditions that you measure are not necessarily the same ones that are needed for that stage of growth and recruitment. So was that something that you put into those, into that portion of the research that you're doing?
So I just want to make sure I understand. So are you saying that you could have like a standing population that's producing seeds, but those seeds can't establish because climate has sort of moved away, and so like the germination niche or regeneration niche has passed by?
Yeah, exactly. And you're saying that like, well, this is the species is persisting, or this population is persisting under these climate conditions, but it's actually not experiencing enough like generation for that to actually persist.
Yeah, I mean, the goal with the collection was to get as broad a sample as possible so we could understand the underlying variation. Your question is a good one about where we see adult trees is very different from where let me start it. Like where you could plant like a seedling and it grow is very different from where you can plant a seed and that tree would grow. And so there's kind of niches that differ between a germinating seedling or a germinating seed and a seedling. And I would suspect that a lot of trees that's going on, especially in the northern hemisphere at the southern end of the range, where the adults will persist. They may even reproduce, but they're not going to produce offspring that are viable. But I don't know that we necessarily need to factor that in directly, because our goal is to identify the relationship between climate and I guess maybe even what you're getting at is like, do we know that these populations are optimally adapted for the environment where we find them? Yeah. And that is a big underlying assumption. Yeah, we're assuming that they're at their adaptive optimum, and they're now getting pushed off of that. And, you know, I go back 100 years, that was probably true before climates really started shifting, but I think that's a pretty safe assumption. Not perfect, but safe. Yeah.
Probably not safe for long. Yeah.
Well, yeah, I mean, I think, you know, we're already pushing them off.
Yeah.
But you know, the genetic information we're sequencing has developed over very long periods of time, and so
Right.
Yeah.
Okay, Dave Secor, please.
Oh, thank you. Can you hear me okay? Yeah, I really liked your climatic analog paper. I enjoyed reading it. And you introduced the idea of novel ecosystems, and I was thinking you'd go right to Hobbes, but you didn't. And when we looked at changing climate way back when in the governor's report, we looked at near sites and we kind of transposed those and we said, oh, South Carolina, Florida, here we come. But I was surprised that you come up with Arkansas and Pakistan as analogs for our region. And the novel ecosystem context, I would think that winters would play a big role in creating novel ecosystems here, that we'll still have severe winters. And in places like Arkansas and Pakistan, they don't. So could you explain how that works?
Yeah, I mean, I'm surprised too, to be honest. Like the Pakistan results surprise me in a lot of ways because they seem too dry for one. But when I've looked at the climate data in that actual grid cell in Pakistan, it's a pretty good match for DC's at least climate as we define it, right? Which is a kind of broad seasonal terms. So there was a second part of your question, Dave, that I lost sight of, and I hope will come back to
Well,
me.
it's just the analog is the counter to the novel ecosystem idea, and winter set the clock for ecosystems,
right.
as Andrew said, and we've seen that you set the clock for what happens the next spring and summer. And is there maybe a factor here where you may have not waited winters sufficiently to get a handle on that?
Gotcha. Right. Okay. So yeah, that's a really important point. Is that what I'm looking at here are cities, right? And the question is, how do you appropriately weight the different dimensions of climate for a city, right? If you're just talking about people's perceptions, you should weight the climate variables to however what aspects of climate people experience most and they associate with that place, right? And that's hard to do when you're looking at a handful of cities versus 500 or 2,000. If you're doing that in a biological system, you can actually do that, right? You can fit a model potentially and say, well, what factors are associated with this system? So I didn't weight the variables in any way. They're all weighted equally. They're all contributing equally. But when we look at what actually drives like non-analogness or novelty, when I've done that, we see that largely you can find very good matches for temperature anywhere on the planet. But what you don't find matches for is the seasonality and precipitation. And so that's getting at kind of like the seasonal thing you're speaking to. And so, you know, I hesitate to say climate novelty is like you're going to Mars or something. It could just be that the seasonal distribution of precipitation is going to change, that San Francisco is going to have rainier summers, whereas right now they have rainy winters or something like that. So that's factoring into.
Thanks. Mark Cochran, please.
Hey, Matt, that was really well done.
Thank you.
So what I'm wondering here is, watching your talk made me remember some of Paul Guger's work where, yeah, I was amazed at one point when he told me how with the oak trees that a tree in one place can actually change which genes are kind of turned on and doing something over time. So I was wondering, you know, whether that might be accounted for in some of your work, or if that might help explain why your random collection is about the same as your selection. And the other part of that would be, if that is in play with these species, whether you might see a different response over time with your common gardens. So you have a certain, you know, result now, but if you wait 5 years, will you have that same kind of result, or will it change? And it has the benefit of saying we need further research so you can go for funding, right?
Yeah, exactly. Yeah, I mean, you're right that like in trees, and I guess, the danger of working in genomics when you've never had a genomics course, even in college, is that you don't have a super deep understanding of the sorts of things like Paul was working on. But, Paul was showing or suggesting that as trees grow, the different age of the tissues can have like different expression of genes and things like that. And so that can contribute to response to environment. So yeah, I mean, we're not factoring that in, and no one really knows yet the role that kind of variation plays, that kind of temporal within organism variation plays. But I'm a big fan of like simple models that we can fit now, because we can't, as much as it's great to find more funding, right? But we can't keep waiting for a better model to be able to inform the decisions we need to make. And so even though I think it's interesting to add more complexity, using the information we have now and going forward with it, I think needs to be done. And then you asked the question about, this random versus selective signal and why. We have some speculative responses, but it doesn't really align with what I would expect. I would have thought that wouldn't have been the case, but we just don't know yet. So more funding.
So Matt, I have a question for you concerning all of the communications work you did after your publication on on this work on climate analogs. I recall that you had scores of interviews on radio, even filming crews coming out to Frostburg. And I know that a lot of scientists, and this is probably particularly a question for some of our early career scientists, who have heard of horror stories of being misquoted, or concerns about how colleagues might think that you're wandering away from your objective science. So looking back on that experience, do you have any regrets or any anecdotal funny stories? Or what would you say to your colleagues at UMCES, and especially the early career scientists, about your experience of being interviewed so much by the popular press and having them try to translate what it was you were trying to present?
Yeah, so I guess I would answer by saying that if I have regrets, it's that I was like so enthusiastic that anybody cared about what I was doing. I said yes to like every single interview. And so that was just like kind of physically and emotionally demanding, like keeping up with all that. And it also means I said yes to people that like I should have never said yes to, like the guy that did the podcast out of his garage and turned into a climate denier like midway through the interview while I was kind of trapped. It actually went quite well, but I sort of realized halfway through that I should have never agreed to talk to this guy. And I should have done my homework first on this person before I agreed. So there's one lesson is, if you get asked to do an interview and you might not know the publication or something, you might want to Google the person and find out. The other super helpful thing was honestly working with Amy Pelzinski and her crew, that they sat me down and did mock interviews and said, like, make sure you write down the main points that you want to get across, because every reporter is going to ask you the question, what did I ask you that you wish I would have asked you? Or what important question didn't I ask you? And sure enough, every single one of them asked me that. It's like journalism 101. And so having an answer to that question and also driving the interview, you really have the ability to do that. And if you say something that you would like to say over, you can always say, you know what, scratch that, I want to try that again. Unless you're live, you can't do that. But I guess that's the major things. Yeah.
Great. Well, I'm glad to hear it was mostly a positive experience, and I'm also glad to hear you talking about the importance of training, and that's something that perhaps we all ought to consider, and maybe that's something we could try to incorporate in some of our retreats and coursework and so on. So I don't see any other questions, and our time is mostly up, and so I guess I would ask those who are on to show your reaction by picking one of those clapping hands, or maybe just present it in the screen like this. And thank you, Matt. I hope you feel like you have a burden off your shoulders and can enjoy the rest of the evening. And thank you, everybody
I
else.
certainly do.
Thank you, everybody else, for tuning in.
Everybody, thanks. It was great.